We're going to start talking about the immune system. And this, in my opinion, is the last of the really challenging parts, I think, or the longest part of the rest of the material in the semester. There's enough content in the immune system lecture that I prepared for you that it will be presented in two different parts, part one and part two. And the first part is going to be pretty much talking about what it is, how the different parts of the immune system were actually discovered, and it's a pretty interesting story at that. And then after we get through all of those sections, I will end the first part and start part two, which is how does stress affect immunity? So having said all of that, let's get started and get our slideshow set up. Excuse me for a moment. a little blank space there. Okay, first, when we're talking about immunity and stress and disease, first we want to think about what immunus, which is the root of immunity, the word immunity means. Basically, immunus, as I have here, is the word for exempt, meaning if you have immunity, you're exempt from having that disease. So immunity is basically a state of protection from infectious disease. Now, psychoneuroimmunology is basically a discipline, if you will, that came together once it became clear that there are interactions between the nervous and the immune systems. And remember, the psycho part is the behavioral part, and the neuro, the brain and nervous system areas, the immunology, obviously, immune system contributions, and all of these three are interconnected. Why are there interactions between these nervous and immune systems? First, the autonomic nervous system projects into tissues that either form or store the cells of the immune system that end up in circulation. And I meant that go and end up in circulation. I've been working hard on this, sorry about that. Tissues of the immune system, secondly, have receptors for pituitary hormones. So if there was not for the fact that there were receptors for pituitary hormones, you wouldn't be able to have this interaction. So as Sapolsky puts it, and again, this is Sapolsky's little saying and not mine, I can't take credit. The brain has vast potential for sticking its nose into the immune system's business. Once again, this interaction, one system is definitely never completely removed or dissociated from any of the other systems. And this interaction is very important. Now, when we're talking about evidence for the brain's influence on the immune system, it was interestingly discovered back a ways that an artificial rose could trigger an allergic response. Now, I love the looks of roses, and I wish I loved the smell of roses, but I don't. And for some people, roses are a trigger for an allergic response. And so the study was done where they tested people and they presented a real rose and then monitored the immune response in the way of an allergic response. And they did this over a time. And then they started presenting an artificial rose. And lo and behold, those same responses, allergic type responses occurred in the presence of the artificial rose. That's pretty powerful. And another line of basically evidence that there is some of this interaction and effect of the brain on the immune system is it has been shown that an actor who had like a day of working on scenes where he experienced euphoria, it was, I always picture like in the old Rocky movies, way back a ways, Rocky finally making it, going to the top, da 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 da, right? And euphoria. And those actors, not just Rocky, but others who spent a day doing scenes in which there was joy, happiness, euphoria, actually showed an increase in immune function and a better immune system in terms of activation under the appropriate circumstances. Now, on the other hand, if it was an actor, and I can think of many movies right off the top of my head, and I won't go through them with you, but I'm sure you can think of some as well. Actors who spent a day or 2 days or 3 days on their depressing scenes, discouraging scenes, depression, actually demonstrate a decreased immune response. And I'm not talking about allergic responses. First, I want to make that very clear. I'm talking about appropriate immune function. So that's pretty strong evidence for this interaction among these systems. Another very more of an experimental, scientifically done study showed if you, let's say a rat, because initially mostly it was rats who were the participants in these studies, were given an immunosuppressive drug, and we know there are plenty of these individuals who have different disorders that are the result of an overactive autoimmune system, take these to suppress their immuno responses. So we know there's plenty of these drugs out there, and so if you give an animal one of these drugs along with a conditioned stimulus. For example, in many of them, in the early days, it was, artificially flavored, during Kool-Aid, if you want to think of it that way, that kind of a thing. So you give them that, and you put the immunosuppressive drug in that. And you can evaluate the immune system's functionality in these rats. There's blood work you can do. There's other things you can do to assess that response. But later, let's not put that immunosuppressive drug in the Kool-Aid anymore. Let's just wait a little while, then give that Kool-Aid again. Guess what? Yes, you're right. The immune system function goes down just like it did initially when the animal was taking the immunosuppressive drug. So the conditioned immunosuppression paradigm. Ader and Cohen, two very important scientists who worked in this area and actually were very instrumental in determining some of the nuances of immunosuppression, studied mice. First, they developed, and you can do this, you can develop a strain where they're genetically going to have overactive immune systems, okay? So here are these mice with overactive immune systems. And Ader and Cohen controlled the disease, the overactive immune systems, with immunosuppressive drugs. Still, don't forget that conditioned part it was a conditioned stimulus. There was a conditioned, like the Kool-Aid or something else that was presented at the same time as the immunosuppressive drugs. And eventually that conditioned stimulus could be substituted for the actual drug and resulted in extending the lifespans of these mice through enhanced immune function. Now, let me back up a minute because I could have said that better and I would fix it, except this is PowerPoint and it's a pain. Enhanced, I don't mean more active in the way that it would become attacking yourself, overactive. I mean a better, more appropriate immune function. So through, maybe not enhanced, but a more appropriate and healthy immune function. So actually, it's no surprise at all that stress can alter immunity. So let's take a look at the historical perspective. I almost said hysterical perspective. Okay, there's some interesting stuff in this little section. At least I think it's interesting. Around 430 BC, in the writings, there was found a description of a plague in Athens, in which only those people who recovered from the plague, said plague, could nurse the sick, because it was found that they didn't contract this disease in a second time. Ta-da, we know what that is now. Back then, they did not know. In the 15th century, Chinese and Turks took dried crusts from smallpox pustules and inhaled them, or inserted them into small cuts in the skin to induce immunity. And this practice was called variolation. And I'm going to ask that on an exam. You just know it. The whole sentence is, for me, a bit repulsive. Okay, let's take smallpox pustules. In the first place, pustules is a kind of a gross thought. But once they've dried, you're going to take the crusts from them, and then you're going to grind them up, and you're going to inhale them. Oh, you say you don't want to inhale them? Okay, let's just make a little cut right here, and I'm going to take some tweezers and stick it in there. Aren't you glad you weren't around right then to be an experimental subject? Next, milkmaids, and this is in the more Western type of country civilizations. Milkmaids were those women who actually took care of the cows, the family cows or a group of cows for somebody else. And they could contract something called cowpox, which is relatively mild. Yeah, you get pox. And you might feel a little yucky, but nothing at all like smallpox. But definitely something kind of in the same family. After they contracted cowpox from their handling of the cow's udders and so on, it was found that they were immune to the smallpox. So in 1798, a guy by the name of Edward Jenner, and I'm pretty sure you've heard of him, injected into an 8-year-old boy the fluid from cowpox pustules. And this reminds me again of back with the effects of like testosterone, and back when there were castrati for the operatic world, how it's like, oh, your young son certainly does sing well. Is it okay if we castrate him? And then we'll always have a lovely soprano. Well, for some reason, parents allow this to happen. Well, in this case, this 8-year-old boy was used for this experiment, completely experimental. And this is where it gets even more interesting. It isn't just, well, let's inoculate him with this fluid from cowpox pustules. Now we're going to give him a little time, and then we're going to go and intentionally expose him to smallpox. Fortunately for that little boy, it worked. He did not get smallpox. This was a very important step in that entire line of experimentation and studies that helped develop not only the vaccine eventually, but also knowledge about the immune system. So let's get into the 1800s, Louis Pasteur. I know you've heard of Louis. And what he did was he kind of worked around and he looked at what Jenner had done with the cowpox inoculation, and he decided, well, let me see if I take this bacterium that causes a disease and weaken it, attenuate it. If something is attenuated, it's weakened. So let's take this attenuated strain of bacterium and let's start injecting it into unsuspecting animals and see if it will protect that person or animal against similar bacteria. And this was actually the beginning of the true vaccine. And interestingly, vaccine comes from the word vacca, which means cow. So when you get a vaccine or a vaccination, it comes from the word that means cow. And he did that in honor of Jenner's work with cowpox inoculation. I always thought this was kind of interesting because, I mean, he could have called it a Jennerine, adenine, something, but no, he went with the vaccine from Vacca. Just my own, probably wayward musings. Now, there is a very deadly disease for livestock, anthrax bacillus. And so moving along the same pathway, and this was mostly under Louis, heat attenuated, because you can weaken with heat, this anthrax bacillus, and then injected some sheep and did not inject other sheep. And then, of course, you know what happened. They exposed them intentionally to anthrax bacillus. And the report is all of the vaccinated sheep lived and none of the unvaccinated sheep did. I'm just going to probably share my own kind of, are you sure, suspicions, or I'm not saying that it wasn't effective. It's just very rare that you have all, none. So I think somebody was very interested. This is just me talking now, okay? Because if I were in class, this is exactly what I would be doing, and I would be getting your feedback. And I'm so sorry I can't do that with this. You have no idea. But I think somebody was very interested in promoting the idea that this was a means to protecting animals, humans. Here we go again with, gosh, what it must have been like to be a young boy during these times. And notice it's always a boy. Louis Pasteur administered the first rabies vaccine to a human named Joseph Meister. And he was just a boy. He was not an older adult who said, sure, come on, inject me with this rabies virus, and let's hope it's weakened enough that I don't get rabies. Rabies is very unpleasant. And it used to be 100% fatal. So Joseph was the basically the experimental kid who was used, and he did not get rabies. They assumed he had immunity to it, but the big question initially is, really, is this attenuated enough? Will this attenuation of the rabies virus protect this individual from actually getting the active full-blown disease? Joseph Meister was so enamored with Louis Pasteur and his entire career of developing vaccinations, of really stepping out into unknown territory and contributing so much to the world in the way of the idea of promoting vaccination as a way to protect people. And it lent more knowledge regarding the actual immune system as well. And a little bit of trivia. In 1940, the Nazis wanted Pasteur's crypt keys. There were crypts that had keys, and it wasn't like you just dig up a crypt, okay? And Joseph Meister actually had the keys to Pasteur's crypt, and the Nazis wanted them. And one can only imagine the things that could potentially have happened had they gotten access to Pasteur's crypt. Joseph Meister wanted to make sure that he did not succumb to their pressure, and so he actually committed suicide so that he wouldn't be tempted or tortured into surrendering the keys. Okay. Bringing up this next section a little bit, there's some of the history, the very beginning of the history through some of the many, many years. And next, along the way, came the discovery of humoral and cellular immunity. Now let's talk about what that means. It was found in around 1890 or so that you could actually transfer the immune state from previously immunized animals to other animals who had not been immunized. This was another incredibly big step. And what we're talking about right here is the serum, or the non-cellular part of the blood, to be very simple. And it was found that the serum contains antibodies. We know about antibodies. We hear about it all the time. We're hearing more about it now than we probably want to. But these antibodies actually can go after toxins and destroy them in one of a number of ways. One, they can neutralize them. They can render them so they just can't do anything anymore. Neutralizing. Think about neutralizing acid. They can precipitate toxins. What does precipitation mean? Well, it means to change the actual state so that it can no longer effectively destroy cells in your body. They could also rupture or lyse. bacteria. They could go right in there and go, you know, with their own little mechanisms. And again, this is not biochemistry class, this is biopsychology of stress. So the important thing is for you to remember these things and have a basic idea of what we're talking about. Rupture, okay, you've got a big goopy balloon, let's say, that's full of bacteria, yuck, 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 yuck. And so now an antibody comes along and buries into it enough that it will rupture. And once the contents are opened up, and this bacteria cell is one that on its own, this one cell can go around and do damage. But if you rupture the cell membrane of this bacteria, it dies. That cell will die, so the bacteria no longer can damage other healthy cells. The fourth way is by clumping the bacteria. If you take bacterial cells and you clump them all together really close, pack them in, they cannot do their job that they wanted to do. They aren't able to go and kill or make sick other healthy cells. The technical term for the clump is agglutinate, okay? The same thing. So antibodies can clump or agglutinate bacteria. Okay, whoops, I didn't mean to do that. Let's go back. So if we, hold on, sorry. I should have taken advantage of Amazon Prime Day and bought myself a new computer, pretty sure. Anyway, because immunity in this case was mediated by antibodies that were contained in body fluids or the humor, it was called humoral immunity. Now, if you took philosophy, intro to philosophy, probably other ones, you probably heard of the 4 humors, you know, and so these were all thought to be like a fluid type of substance. So this was called humoral immunity. Now, phagocytes were discovered around 1883, and they were found to be cells who could ingest, basically eat, microorganisms and other foreign material floating around in there. And it was found that these were more active in animals who had been immunized than in those who had not been immunized. So these are cells, right? Cyte means cell. These were cells. So now the hypothesis emerged that it was cell-mediated immunity that is important. And next was the lymphocyte identified. Look at this, not until the 1950s. And when I first started, you know, teaching this kind of material, it seemed like so recent. I mean, not that it was like in the 1950s I started, trust me. But in the whole scheme of medical history, it seemed like in the 1990s, it seemed very recent. Now it seems a little more distant, but if you consider discovery of various medical and physiological mechanisms, I think you will understand that it is pretty recent. This cell was identified, and then it was found to be responsible for, guess what? Both cellular and humoral immunities. Wow, imagine that. And just as happens so often, and it's really kind of sad, because I think you know, I just wish there could be this, oh, maybe there is both. But for a while there was, it's all about humoral immunity. No, it's all about cell-mediated immunity. It turns out both of you are right, and they both are important. And in fact, we have a cell that is responsible for both cellular and humoral immunity. Okay. Next, let's take a look at the various components of the immune system. I'm not sure if you remember back when we were talking about the cardiovascular system, and I mentioned the lymphatic systems, and I said that we would be talking more about that later in more detail. Later is now. So the lymphatic system basically includes vessels that transport lymph from interstitial spaces to the blood. So remember back the lymph, and there's what's between the cells. It's outside of the cardiovascular system particularly. And then we have these little tiny vessels that can take this in the fluid from between the cells, the interstitial spaces, to the bloodstream. Lymph basically is what is left of the interstitial fluid after the rest returns to the blood in the circulatory system. Just another reminder, we talked about how some of these cell walls have gaps just so fluids can be extruded through them, and fluids can be returned to the blood via the circulatory system. I have just inserted a little diagram for you here that shows different components of the lymphatic system, and all of these different structures and the vessels associated with them are very important in our immunity. That's this diagram. So let's talk about what some of these are. First, primary lymphoid organs. And bone marrow is probably not what you would first think of, but it is. B cells, cells that are very important in immunity and this whole system, are born in the bone marrow, and they also grow up in the bone marrow. And it means that they are born and then they develop and mature, and then they can circulate through the bloodstream. T cells, and I know you've heard about T cells, originate in the bone marrow. They're born there, but they don't mature there. And we're going to talk about that a little bit more in a few minutes. So the thymus is a flat bilobed organ above the heart. You can see it in that diagram. And immature T cells that were born, that originated in the bone marrow, actually migrate to the thymus, and they're supposed to mature there. And at this point, before they've matured to the full-blown T cells, they're called thymocytes. Makes perfect sense. Again, this is how I would have named them, because it's thyme. Oh, that makes sense. Oh, cyte, that's a cell. Sounds awesome. Now you go, okay, that's great. And it's, I can't help it, the way my mind kind of associates different things. The commercials where they were trying to decide whether cheese was mature enough to be used to make cheese cracker type things. And they, big glob of cheese would come in and they'd ask them questions, and it would be silly and immature, and they go, not yet, you're not ready yet. Well, once these thymocytes are within the thymus, 95 to 99% undergo programmed cell death. That's crazy. Think about it. It's like if you in this class, you got here and you got into Canisius and you're at Canisius and you're, You're like gonna graduate, at least you're thinking, but you don't mature enough. And sorry guys, program, you didn't make it. That's what actually happens. And again, down the road here a little bit, I will explain why that happens. It's actually a good thing. So among the secondary lymphoid organs, we just finished primary, now we're to secondary, are the lymph nodes. And I'm sure all of you are very familiar with lymphoid organs as well. And I mean the lymph nodes, they're encapsulated bean-shaped structures. And if you've had a bad sore throat or tonsillitis or anything like that, you might have had them down in your neck. You could feel them along the side. Other places, sometimes you can feel them in your armpit, through there, in your groin. But what is in them? What is in these bean-shaped structures? Lymphocytes and macrophages. They actually are packed with these cells. And lymph, remember we just talked about that a minute ago, percolates through these nodes. They're not forced through with pressure from arterial constriction or anything like that. They percolate through. And when they do that, antigens, antibody generating molecules, are trapped there. Don't worry about the antigen too much. We're going to talk about that as well. Just trying to give the overview right now, and then we'll talk a little bit more specifically about some of these things. Now, the spleen is a large ovoid organ high in the abdominal cavity towards the left side. And basically the spleen filters blood and the antigens that are carried in the blood. So the spleen does a different job than the lymph nodes. The lymph nodes basically trap antigens in the lymph, and the spleen traps antigens in the blood. Very important difference. Now, there are two broad categories of immunity. The first we're going to talk about is the innate or non-specific. It's important that you connect those two terms. This immunity is the basic resistance to a disease that an individual is actually born with. And there are defensive barriers, 4 of them, that constitute this basic resistance, this innate immunity. The first category is anatomic. And I guarantee, this isn't even a 99.999% sure possibility, it's 100%, that you will need to know this for the final exam. And, quiz maybe beforehand or something. So anyway, go away you. Get rid of you there, thank you. And move you around a little. Skin is the first barrier. The skin keeps many, many things from just going from the air into your physiological self inside your body, into your bloodstream, into an organ, into tissues that are just underneath your skin. Mucous membranes in the mouth, anywhere in the nose, there are mucous membranes in different orifices, if you will, and they are an effective barrier to the entry of most organisms if they're healthy mucous membranes. The second category is physiologic. So we are physiological, you can say either. And so the first one is anatomic, the second is physiologic. Temperature is one of the ways that we have this physiologic barrier or part of our innate system. We're so kind of, I think, entrained to, oh my gosh, you have a fever. You better take something and bring it down. Well, that's true if your fever is too great, if your temperature gets too high. But the whole purpose of mounting a pyrogenic response or a fever is that higher temperatures can kill proteins and other things that are dangerous to you. So it's kind of like if it gets too high where it's going to damage really important cells and just not the invaders, then you need to bring that temperature down. But if you have a temperature of 100, don't be, it's really not a great idea, at least in my opinion, and the opinion of many other people with whom I've spoken about this. Let a little fever do its job. pH is a second of these like little actions that can be important for this category of innate immunity. The low pH, acidic environment of the stomach kills most ingested invaders. Now think back to when we were talking about the digestive tract and all those things, that this is good for in the stomach, but once you move past that, you can't have all that acid. So it needs to be neutralized with bicarbonate. Third, chemical mediators. There are lysozymes. Once again, lyse, split, rupture, breakup. Lysozymes, these are little mediators that cleave bacterial cell walls. So they can go in and it's like, I'm cutting you open, bacterial cell wall. And once that cell wall is destroyed, then that the interior of the cell cannot stay intact and healthy. Thus, it will no longer be a healthy active cell and will fizzle out. Very technical term, I know, fizzle. Anyway, interferon. An interferon induces an antiviral state in uninfected cells. And the one thing about viruses is they need to go in and take over a cell. They go in and it's like a little alien and gets into you and then makes you do something different. So what we need to do is design a way that will keep the virus from basically being able to get in and take over the cell. That would be the antiviral state. Also, complement. Notice this is not compliment, not like, oh, I love your dress. Oh, awesome hat. That's a compliment. This is complement. And really a complement is a group of serum proteins that actually aid in damaging or completely destroying a pathogen or pathogens. So third category in these four categories of innate immunity, endocytotic and phagocytotic mechanisms. Now you see the site, so you know it's cells in there. Endocytosis is the ingestion of extracellular macromolecules. So this is one of the ways in which our innate immune system can help destroy molecules that can be damaging or dangerous to us. Phagocytosis is the ingestion, think of a little Pac-Man, of whole pathogenic microorganisms. So the difference is kind of the size, if you will, of the actual substance that's being, consumed. And final and fourth inflammatory barriers is tissue damage. If you damage tissue, it induces three major events. One is vasodilation, basically opening up vessels. An increase in diameter of blood vessels occurs as vessels carrying blood away constrict. And the capillary network is engorged. That's a great word, engorged. That's really like so much better than filled up. And so this results in redness and increased temperature. I can't imagine there's many of you who haven't experienced that, where you have damaged something, you've injured yourself, and that area becomes hot, it gets red, and then you start watching because like, is this getting infected? And you start watching that area. And another inflammatory barrier is an increase in capillary permeability, things that are able to get through. And with this, we have an increased influx of fluid and cells from the capillaries into the damaged tissues. This causes tissue swelling. I think it's kind of easy to visualize that, something that we would call edema. There is an influx of phagocytes. Remember the phagocytosis we talked about just a minute ago? From the capillaries into the tissues. And accumulation of these phagocytes leads to the release of lytic enzymes. Lytic, lyse, chopping up, breaking apart, enzymes that can actually end up damaging healthy cells. If you put this stuff together, you end up with dead cells hanging out, digested materials clumped there, and fluid, and that is what becomes pus. Another one of those words that sounds like what it is, like mucus, pus. Anyway. A principal mediator of the inflammatory response is histamine. Now, this should make sense, I hope. Histamine is released by a variety of cells, okay? So we're not getting all fussy and picky about that. But histamine binds to receptors that are located on capillaries and venules, and it causes vasodilation and increased permeability. So there more fluid can kind of flow back and forth across the vessel. And before I go on to the acquired immunity, I want to mention about histamine. That's why if you're getting hay fever, you're all stuffy and swollen in your sinuses and your eyes are getting swollen because of an allergy or something, it's because it's histamine. You take an antihistamine, and what that drug will do is block that histamine and will then reduce all of this swelling and extra fluid and maybe even redness and everything. So that is the principle behind antihistamines, very briefly. Now, we've been talking about innate immunity. That immunity that you are born with. It is like a built-in, but it's not specific. It is a general type of immunity. Now, acquired or specific immunity is the presence of a functional immune system that is capable of specifically recognizing and then selectively eliminating foreign microorganisms and molecules. And there are 4 attributes to acquired immunity. Just like I talked about the 4 different categories of innate immunity functionality. So the 4 attributes is, first of them is 1, antigenic specificity. Now, I think I had this on a separate little document, and I've got to make sure I include it, but it's a few definitions about the immune system. And antigen is what comes in and is foreign that your antibodies need to recognize. Antigen, okay? Antibody generating cells. Now, our immune system, our acquired immune system, if it's healthy and functioning correctly, can distinguish subtle differences among antigens within a single amino acid. We are so good. Second attribute, diversity. Not only does a system have antigenic specificity, able to distinguish subtle differences, but it can recognize billions of unique structures in foreign antigens. Third, acquired specific immunity exhibits immunologic memory. Okay, so let's say you have an encounter with this foreign material and you're like, oh, I'm going to attack you and get you out because you're not going to be very good for my body, for my human's body. And then, so you get rid of it. You might actually, as part of this immunity, now I'm kind of acting like you're this system. So stay with me for a minute. And you're like, phew, got that over. Man, he made it in so far and he could have damaged so much. In the classroom, face to face, used to be nice. I kind of use the analogy of somebody kind of coming into the classroom, long dark trench coat, but we're like, well, you know, it's a little rainy out there. He comes in, sits kind of in the back. I'm like, not on my roster, but doesn't seem to be hurting anything yet. I don't recognize this person. And then as the bell, we don't have a bell, as the class ends, goes up the aisle, grabs somebody's laptop and runs out with it. And it's like, whoa, come back. Oh, bad you, and it's like, man, we're not going to let that happen again. So a couple of weeks go by, this guy comes in again and he's like, we're like, you look awfully familiar. And kind of thinking about, but just before we can go and hold him down, before he snags somebody else's laptop, he gets it and goes. And we're like, oh man, we got to be ready. We've got to recognize this guy the minute he comes in the door, and we are going to mount our own attack. So the next time, everybody gets up and like, oh no, you leave now, we're getting rid of you. And that memory, and then a heightened state of reactivity. Okay. Now, fourth and final attribute is this specific acquired immunity is capable, it should be, and this is a problem sometimes, capable of self and non-self recognition. It is supposed to only respond to non-self molecules. That is to keep your immune system from eating up your own tissues. Now we know there are autoimmune diseases, and unfortunately it seems like they're occurring more and more, whereas your immune system starts attacking you. Autoimmune. And that's because there is a problem with this self and non-self recognition. So actions are triggered by cells that belong and not just foreign ones. Okay, I'm gonna take a little breath for a minute here. I really want to get through this section. So after that, we can turn in a separate video lecture to what stress does to the immune system. So take a breath and let's move on and talk about the cells of the immune system. We have talked about some of them as we've gone through, but I'm going to just come up this far. Okay, white blood cells, leukocytes, think of leukemia, are attracted by chemicals released from damaged cells. And this is called positive chemotaxis. So these white blood cells are like, oh, I feel like there might be some damaged cells over here. I'm moving towards it. Think of a taxi, chemo because it's chemical, positive because it's going to and not away. And these guys can squeeze between the cells of blood vessel walls. And this going through, this squeezing through, and I've got to eventually get a whiteboard here, I guess. If you look at the different cells, and then there's that little bit of space in between that I've talked about many times in terms of blood vessel walls, it can squeeze through. And the word for that is diapedesis. Dia literally means through. And I know you're probably like, oh my gosh, here she goes again with a Greek or Latin language lesson. I just want to share it with you because I am telling you that if you know what some of these roots and prefixes and suffixes mean, you can figure out the meaning of words you've never seen before. And this is one of that. If you know dia means through, diarrhea, diarrhea, flow, rhea, through, dia. Diapedesis, think about little feet going through. I'm walking you through, okay? So that is something that can happen. Now there's two main types, granulocytes and agranulocytes. Cells with granules and without. That's so basic and simple and actually way too simplistic in a way, but it is true, and that's all you need to know. Of the granulocytes, we have these: basophils, eosinophil, and neutrophils. If any of you have noticed some of the commercials for this new treatment to add on for severe asthma that's caused by eosinophils, that's what we're talking about. Again, we want some, we just don't want too many. Too many, again, starts to become like a situation where overdoing the immune system. The second, the agranulocytes include lymphocytes and monocytes, and those are macrophages. So for a minute, let's just take a look at the monocytes, which are like phagocytes, macrophages, and dendritic monocytes. There's two different types. These, and this is kind of what I want you to know for this section, the largest cells in the blood. They are 2 to 3 times the size of red blood cells, and they live for several weeks or months. So they hang around for quite a while. They're very mobile. They can engulf, and again, I just picture Pac-Man. Relatively large objects, and I mean objects, not like a dog or cat or chair or table, just a little whatever can be carried around in your blood. And they can kill, and this is an interesting thing, it's good to know this, can kill invading cells by producing large amounts of nitric oxide. And nitric oxide, just as an aside, is an endogenous gas that's released. I actually studied it a bit when I was working on my PhD because dopamine release is triggered in part in some areas of the brain by nitric oxide synthase and that whole thing. But we need some, we don't need too much. And one of the things that has been found in stroke victims is that the effects of that stroke start causing, the inflammation and, damage to surrounding cells. And then you suddenly have more nitric oxide being released. And that then will kill cells. Again, we can overdo things. And when I say we, I'm talking about our immune system. Hopefully you got that. Well, we can overdo things too, I suppose. Eventually, these monocytes are like giving themselves up to save you because they become so engorged with digestive products and bacterial toxins that they die. Next on our list are lymphocytes. They're only slightly larger than red blood cells, so much smaller than the monocytes. They may actually live for years though, so longer living. And they originate as stem cells in red bone marrow. Remember bone marrow? Before birth. Now, I'm frustrated because there was no way I found out that I couldn't bring both sides up at once. So this is what we're going to have to do. They originate as stem cells in the red bone marrow before birth, and about half of these cells reach the thymus glands. They're thymocytes, remember? And there they differentiate or mature into our T cells. And that stands for thymus-derived lymphocytes. And here's where we go back to why many of them don't make it out. They are educated not to attack self. I'm still going down on the left-hand side here, 90% of them die because they recognize self proteins, not just foreign proteins. What does that mean? It means they will attack self. Autoimmune. So if so many of them don't learn the difference, if they don't get to a certain point, they die off, and that is supposed to reduce, and it does reduce the chance of autoimmune diseases. Then, and I will come back over to the right side in a minute, still going down the left. They are transported to the blood, to various organs of the lymphatic system. And then you've got cytotoxic T cells, which attach to bacteria, antigen-bearing cells, and interact directly. Remember that cell-mediated immunity. T cells can detect antigens on virus-infected cells too, disrupt their membranes, destroying both the cells and the viruses they contain. T cells may also secrete toxic substances, lymphotoxins, that are lethal to their target cells. That's a good thing. They also may secrete growth inhibiting factors that may prevent target cell growth. Target cells being those invading cells. You're going to target them and get rid of them. Or they may secrete interferon that prevents the proliferation of viruses. T cells and other certain cells, such as macrophages, can synthesize and secrete cytokines. I have talked about these before. Some people say cytokines, potato, although I've really not heard anybody say potato, that's an aside, which enhance various cellular responses to antigens. So here we see another example of synergistic activity. They can enhance various cellular responses to the antigens. However, it is very important. These do not release antibodies. They do not. Examples of cytokines are interleukin-1 and interleukin-2. Sometimes you'll see that simply as capital I, capital L1, capital I, capital L2. And these can stimulate synthesis of other cytokines, both of the interleukin-1 and interleukin-2. They can activate resting T cells that are there, they've matured, but they're resting. They're not engaged in any battle at the time. And they can also cause a proliferation of T cells and activate cytotoxic T cells. And I have some links here for you, but before we can even talk about that, I need to go back. Sorry, I couldn't be more efficient with this, but I worked at it a little bit and then I'm like, finally, it's like, we'll get through it. So thank you for your patience. Okay. All right. The stem cells originating in red bone marrow that don't reach the thymus gland, that don't go to the thymus gland, the school of learning, immunity, and self-recognition and other recognition, those will differentiate into B cells. And that simply stands for bone marrow derived lymphocytes. And they become either plasma cells. Here we go, antibodies now. These plasma cells secrete immunoglobulins called antibodies, IgG, IgE, and they are carried in body fluids, humoral. Or memory cells, which lie in waiting until the next invader comes along. And that is directly humoral immunity. Here, these antibodies are carried in body fluids, but they are the cells themselves, so it's cellular immunity, okay? And that basically brings us to the end of part 1. And here are some different links. First couple aren't very long. The third one's a little bit more detailed, but I encourage you to watch at least a little bit of it. I'm not going to take time here and show it. That's something that definitely you are able to do on your own. And so I encourage you to do that before we move on to the next part of the immunity and stress. And again, the reason I moved the immune chapter to after the first exam was because it is very substantial. It's meaty, it has more information than some of the others in. So I thought it was better to like break it up and do it after the first written exam. So you know it's going to be on the second written exam, and getting it now gives you an opportunity to get familiarized with the material and take your time with it. And then after that, believe it or not, at least in my opinion, things start getting easier. And so hopefully as the end of the semester gets more tense in some ways, this should not be like that. I'm hoping anyway. So thanks so much for your attention, and I will see you soon when I start